It's my pleasure today to host Rachel O'Neill from University of Connecticut. So Rachel got her PhD in Australia, studying comparative genomics, and then did postdocs at Princeton and Rutgers, and has been at UConn for how long? I forgot to look. 23 years. So I think what's really characterized Rachel's work is that she has worked on the hard parts of the genome for much longer than it has been fashionable.
It's very true.
So, lately people have started studying repetitive sequences, but 20 years ago it was really, really hard to do it, and that's what Rachel was doing. So her lab has done a huge amount of work on transposons and simple sequence repeats, and have worked on some weird animals like the Tamer wallaby and other marsupials. Maybe that was just an excuse to get to do field work in Australia, I'm not sure. And then recently she's been part of the telomere to telomere genome consortium, where a large group of labs have gotten together to actually finish the genome. So sequence all the way through the centromeres and the repetitive elements in the rDNA, although I think the rDNA is a little bit more of a challenge than the centromere. So I think she's going to tell us about a lot of her interesting work today. So thanks, Rachel.
I'll take you on a little bit of a journey across many things. Thank you very much. And I apologize, I was supposed to be here in the spring, but between a snowstorm and blowing out my knee, things got delayed a little bit. So thanks for coming today. So back in 2000, Y2K happened. The world did not end, but we did have notification that a really exciting breakthrough had happened, and that was that a human genome had been completed, or at least that's what we thought at the time, right? And this was celebrated, and really at the time it was a massive feat. It was very expensive. It was $2.7 billion. It took hundreds and hundreds and hundreds of scientists and hundreds of thousands of hours of effort. So it really was a great feat. But what that genome looked like at that time was not complete, right? Even though we said we completed this genome. So this is what the human reference looked like in 2000. And you can see there's some pretty major gaps in there where there's no sequence at all. And the vast majority of this genome was less than 50% confidence, right? And that's what I'm showing you here in yellow. So what that means is it might be a C, it could be a G, right? You had a 50-50 chance of that being accurate. So this serving as a reference for clinical studies was quite challenging. So over time, they continued to fill these in. This was using technology like back-end sequencing, etc. And you can see here, we're increasing the confidence calls of many of these chromosomes. And that's what we're showing you here in black. Things that were or in pink, things that were unchanged are here in black, but those gaps remained, right? And they're scattered all throughout the genome. And over the next few years, we had some incremental updates. So this is in 2003. Some chromosomes got new coverage, like 1 through 5 and 12. Some chromosomes, like 13, 14, 19 through 22, saw very little, if any, change at all. And the Y chromosome was just kind of left here. And this is pretty much the story over the next few years. So in 2006, less than 1% of the genome flipped its confidence scores, and none of the gaps were touched. And in fact, in 2013, the last time the human genome reference was updated at this scale, this is GRCH38, is 2013, 10 years ago, about 8% of the genome was not sequenced at all. So we still had a lot of these huge gaps. We had some really low confidence regions, particularly surrounding many of these gaps. And then these two guys came along. This is Adam Philippi and Karen Miga, and they had this idea, we really need to tackle the human reference. It is still missing 8%.
Just to interrupt, your
Is it dead?
speakers, the battery's going in and out. So.
$50-50 chance. That's better. All right.
See that one there?
All right. Can you guys hear me now? All right. So these guys came along and said, you know what, we're going to complete the human genome. And we're going to use modern technologies to do it, but we're going to do this in kind of a different way. We're going to do this in an open science and fully collaborative way. That meant that anybody that wanted to participate could participate. It also meant it was not funded. Which is kind of a key point, right? This was a completely unfunded project. And so we joined the team with the intent of just giving a little bit of data, some pro-seq data and some repeat data. And our participation in this team actually grew. We were lucky enough to be one of the 6 papers that came out with this consortium. But it really was a celebration of a new way of doing science and a really exciting way of doing science. And so what came out of that was finally, we actually have a human genome that's completed. All of the gaps have now been filled in. And it's a truly gapless genome. And we call it a human genome because it's just the start. Now the initiative is to do hundreds or thousands of genomes so we can get a really good reference of what the human population looks like. So why was this important? For my work, this was incredibly important because each of these gaps happens to be a centromere, which is an area of the genome that I was quite interested in for evolutionary reasons. And we had no human reference for a single chromosome, right? And it's kind of surprising when we consider how important centromeres are. So they are the site of kinetochore attachment, spindle attachment and kinetochore assembly. Without a functioning centromere, you don't get a spindle attachment, you don't actually get propagation of that chromosome, and the chromosome is lost. And we know this is guided by a centromere-specific histone known as SEMP-A. How it knows where to go in the genome is still something of a mystery. Right? We don't know what the genetic determinants are. We know there are epigenetic features that are involved in that deposition, and I'll talk about that in a second. It's functionally conserved in that SEMP-A foreign centromeric chromatin in a wide range of species. Right? And this is conserved from humans to yeast. Right? But it is sequence divergent. That means no 2 species actually have exactly the same centromere sequence. It's very rapidly evolving. And in fact, it's the most rapidly evolving sequence in our own genomes. And we can see they also, the repeats that are found within centromeres form very complex genomic structures. So for example, in human, we have satellites, alpha satellites, which are about 180 bases, and they're tandemly arrayed for megabases of sequences. But what happens is any 2 of these satellites may not be that similar to one another, 70–80% identity. But a block of those will then be repeated. And that's what we call a higher order repeat structure. And this is where homogenization starts to happen. So this particular satellite here is almost 100% identical to this satellite. So this makes assembly across these regions very challenging. This is one of the reasons why these regions were the last to be assembled. It's a bit like thinking of a puzzle that you want to put together that's a picture of the Sahara Desert. If that puzzle was 10 million pieces, you'd have just a lot of pictures of sand, right? And it'd be very hard to put together. But technology has changed such that we can turn that 10 million, 10,000, 1,000 piece puzzle into a 10-piece puzzle, right? And that's allowing us to do these assemblies. But what we also know is that the forms of those centromeres can vary. Even within humans, we can find that we have mobile element insertions. In many species, we have emerging centromeres that have very few and some that have no satellites. So there's no one rule that applies to all centromeres except the SEMP-A rule. We also know that it's not necessarily fixed in the karyotype. So that means that it can form neocentromeres, and we see this in clinical cases. We also see this in evolution. We can have centromere shifts, is what they're called in an evolutionary context, where there's no change in gene order, but a centromere just moves from one location to another, right? Just one's off and another one's on. We can also get fissions and fusions and whole arm reciprocal translocations, so major chromosome rearrangements that all seem to happen within the centromere. So it's a very dynamic part of our genome, as well as genomes across evolutionary time. And what we entered the T2T consortium to do was just, we wanted to know, are there undiscovered repeats in the human genome? And in particular, are there any in those centromeres? And we had a really strong focus on transposable elements. We wanted to know if there was any relationship between TEs and satellites. And a lot of that centered around the idea of transcription, because there was quite a bit of literature showing that satellites could be transcribed, but the interesting thing is they have no promoters, right? So maybe it's TEs that are doing some of this work, we don't know. And then more importantly for me in my career is, can T2T level comparative genomics really reveal how centromeres function and evolve over time? And that's because if we look at different species, we see many different forms of centromeres. We don't quite know how one is related to another. So for example, in human, we may have these dominant satellite forms with transcriptionally active elements that may sit in the middle, or we have neocentromeres in clinical cases, or evolutionary new centromeres like we have in horses. We can get complex centromeres that start to accrue the same repeats, and then these more stabilized centromere forms. So how are each of these related to one another, and are they actually a cycle? And I'll talk a little bit towards the end about two model systems that we're using to sort of address that problem. One are the marsupials that we heard about a minute ago, and the other is another model system that I'm actually in love with, which are the gibbons, which is a non-human primate. All right, so back to the telomere to telomere project. How was this achieved? When Adam Philippe's group first decided we're gonna do this genome, at that time doing a diploid assembly was not possible, right? So being able to resolve one haplotype from another just made assembly errors higher. So what they decided to do was, instead of taking a normal diploid cell line, they took a hydatidiform mole cell line, which is CHM13. It's 46XX, but both chromosomes are paternally derived. All right, so this is an androgenetic form. It is a failure. This is a pregnancy that fails. Molar pregnancies do fail, but it becomes diploid in a haploid genome, right? So it makes the computational aspects much easier. And Adam's team built this assembly from a high-resolution string graphs using PacBio hi-fi reads, which were the prominent and very high accuracy reads at the time, and manually resolved many of the tangles that they found using what was an emerging technology, which was Oxford Nanopore ultralong reads. And at the time that they were doing this, ultralong meant 20 to 40 kb, right? And that has changed dramatically in the last 3 years. Now we can get ultralong reads that are in the 1 to 2 megabase range. And so what this allows us to do is to go from having to use an androgenetic line like this to being able to actually phase haplotypes. And so a way to conceptually think about this is, maybe you have a paternal and maternal allele that differ here, or the same here, differ here, or the same here. How do you actually build an assembly using that without, having 2 copies of this and 1 copy of this, right? How do you get rid of haplotypes but represent them in the assembly? These ultra-long reads allow you to do that, because that allows you to take each of these unique sequences and make a path along each allele, right? So this is now how we're deriving diploid assemblies. And what that ultralong data allows us to do is take an assembly graph that looks like this. So this is an assembly we did back about this time that telomere to telomere project was starting. This is actually a marsupial genome. And now it looks like this, just by adding this ultra-long data. And so each of these lines is a chromosome. So we're getting single chromosome resolution. So it's an extraordinary time to be a genome biologist. So what our team was involved in with the T2T project, we actually thought we were just doing some, look-sees and participating in the centromere team. And then all of a sudden one day somebody said, we've actually added all of this genome sequence. We need to completely redo the repeat annotations because we don't know what's in there. And the way repeat annotations work is people tend to use a database which has known models in it to annotate a genome. So if there's new repeats, they're not in that database. So 3 grad students in my lab and a postdoc, all of these guys have actually are now postdocs in my lab. I'm very excited about, led this effort where we were assigned to fully annotate the genome. And that meant all simple and low complexity repeats, satellites and satellite variants, and anything that we found that was an alpha satellite or in the centromeric region, we passed over to what was called the SynSat team, led by Karen Miga and Nick Altimos and Ivan Alexandrov. And they actually did some really deep scale phylogenetic analysis, superfamilies and this kind of thing with the centromeric sequence, which is really exciting. It's a great paper, you should check it out. And then we also looked at composite and complex repeats, repeats that had multiple repeats within them and then they were repeated. And we also targeted interspersed repeats. So this includes all retro elements and class 2 elements, including DNA transposons, structural RNAs, tRNAs, you name it. And what that meant is we annotated 53% of the genome. And of course, we were bringing in the tail end, right? So everybody else had already done their annotations. We needed the gene annotations to be done before we could do any of these repeat annotations. And so I always laugh because the genome annotation team did an amazing work on the gene annotations. We discovered up to 2,000 new genes in the human genome, 100 of which are probably protein coding, but it was 2% of the genome. And then they're like, "Oh, do the repeats." And we're like, "It's like 53% of the genome." So it was a huge effort that this team and a whole group of scientists actually participated in. And so what we did was we developed a new a pipeline, and we Jessica Storer at the time was in Arian Smith's lab, and Daniel Olson was a grad student in Travis Wheeler's lab, and they all came together and said, "We have this novel new way. Let's try this and see if this will work with this genome." What was exciting is that everybody that was involved in leading this project were trainees. And that's a note I should make about the 6 papers. All but one were first authored by graduate students, right? So this really was an open, collaborative effort. So we masked the genome, and then we did a novel modeler. So by blacking out every known repeat, we then asked, are there new repeats that are in there? We then collapsed those, merged them, and through a very long process did a bunch of manual curation. And I'm not going to go into the gory details. Anybody that wants to know about it, call me. We are happy to share the pipeline with anybody and everybody. The manual curation part, nobody will ever have to do for the human genome again, but we are having to do it for other genomes. And we spent a lot of time doing this so no one would have to do it. So we would build the most comprehensive database we could. And so all of the repeats now are in DFAM. And so the manual curation involved intersecting it with genes and retrogenes to make sure we weren't calling retrogenes that may be repetitive a repeat. Looking at segmental duplications and being able to annotate those appropriately. And then anything that may be a low Smith-Waterman score for an alignment for making a consensus sequence. Like, is it just an old repeat? Is it a young repeat? Do we need to set it aside? And then the thing that we also discovered is repeat masker and repeat modeler are terrible at tandem arrays. We would be skating along the genome, and then there would be a gap of 50 kb that had no annotations of any kind in it. No genes, no repeats, nothing. And so this is where Daniel Olson came in. He designed a method called Ultra that's better than tandem repeat finder, but it required a lot of manual curation, just looking at these stretches of DNA, and we would find that they would be satellite arrays, unknown satellite arrays. And then taking all of that and collapsing it into one annotation file that would be available for the reference. And that's what we derived here with improved calls, satellite arrays that were newly discovered, and composite elements. And so what that looks like, if we compare CHM13 to GRCH38 and just what repeats are lifted over, so which ones are shared between the two. About 17,000 were not lifted over for TE annotations. What does that mean? So that is 7.6 megabases of new TE annotations in CHM13. So it's actually a lot of new transposable element annotations. Interestingly, and quite surprisingly, about 80% of those were in syntenic regions. So one of two things is possible. So what that means is the exact same region in GRCH38 and in CHM13, now there's a line element, for example, in there. And the question is, so it wasn't in a gap. So this is not a gap region. The question is that because this collapsed that line element out of the assembly? That's very possible. Or is this a polymorphism in the human population? And so now this is the challenge for the next phase of GRC, or of HPRC, which is the Human Pan Reference Consortium. Of the ones that did lift over that were the same, many of them actually got reclassified because we had better sequence data and better confidence in the sequence types. And because we had a complete reference database, we could actually build accurate consensus models for every repeat type. For new repeat annotations, so of those, about 119,000 TEs were corrected. For new repeat annotations, which I thought was the cool and exciting part, we have 64 new human repeats that have never been seen before that we've now annotated. And that encompasses about 18,000 new repeat annotations in the genome, which is about 5.5 megabases of DNA. What was kind of surprising is that they were all over the karyotype. So we had many repeats. There were new annotations within centromeres, and I'm showing you here in black on the edges are the gaps. You can see the gaps are not all centromeric, but they are scattered throughout. But some were in regions that weren't gaps. And so we were like, why is that? Why would that happen? Some were just repeat modeler gaps, but many were repeats that were only found in one copy here, but many copies in the centromere. And because the centromere wasn't resolved, they weren't classified as a repeat. So we now have better resolution on eukaryotic regions of the genome because we have resolution of heterochromatic regions. And that's a really important aspect of this work. So one example of a collapsed repeat that we found are what are called composite repeats. And we found 19 of these that span 2.8 megabases. And so what this is a repeat that consists of multiple repeats in a block that's tandemly arrayed, right? And some of these, for example, this acro repeat, we find a huge block of them on chromosome 22, may actually be found in, and that's this array here. And you can find many arrays here, 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 and here. You can find some in the centromeres, but then we also find single copies of this block in other portions of the genome. Everywhere that it was arrayed was either a gap or it had been collapsed to a single copy. Right? So the way the sequencing technology was at the time that the first reference came out couldn't actually resolve that there were tandem arrays of these. And now that we have long read data, we can actually count exactly how many copies we had. Why this is important is that some of these actually contain protein coding genes, such as the gauge locus that's on chromosome X. We've now got 19.2 copies on this particular X chromosome, and many of these had actually been identified by Peter Warburton's work earlier on when he looked at large tandemly arrayed gene families. So now we've actually been able to define every repeat that's within them and what the boundaries are. So these weren't necessarily our novel discovery, but now we have better resolution. So one repeat I do want to give you a little bit of a sidebar on is a repeat called SST1. And this is a transposable element derived repeat. It is found in big arrays on chromosome 19 is the one that has been known, and chromosome 4. Prior to CHM13, we had no idea there were copies of SST1 anywhere else in the genome. And this particular repeat is really important and interesting because at the time, we knew it was overexpressed in certain types of cancer cells, right? But knowing which locus was actually contributing to that expression was impossible because we didn't actually know where all the copies were. So we've now annotated the copies all throughout the genome and then pulled out every single copy and did a massive phylogenetic analysis. And so what we did, and this is just a collapsed tree, just showing you the relationship. So we wanted to know whether the arrays on 19 and 4 were progenitors of all these other sequences or how they were related to one another in any way, shape, or form, right? And what we found in this tree is that, in fact, chromosome 19 is probably the original sequence that then propagated across the karyotype. How it did so is unknown. No idea how these arrays propagated to other places in the genome. But because of the type of DNA sequence that we had for this genome project, we were able to derive DNA methylation calls, and this is work by Winston Timp's lab. And we were able, by work in my own lab, to look at ProSeq. So we were able to look at precision run-on sequencing to see where active RNA polymerase occupancy was. And then we were able to add this data on a base-by-base method to every single copy of SST1 across the genome. So we had an unprecedented detail of epigenetic and transcriptional features of every single one of those elements. And what we found that was really interesting is this little block down here. And what you can see here is that it's highly methylated. The darker is high methylation. And contrary to what we would predict, highly expressed. In fact, off the charts expression. You can see 600,000 reads, right? So really high expression levels. And it just so happens that it's these chromosome 19 and chromosome 4 arrays that we had talked about before. Very highly expressed. And so we took every single copy of all of these elements and put them on a graph like this. And so each line is a specific sequence. And what we're showing you here is what the pro-seq density is. And so this allows you to figure out if you have promoters, for example. And what you can see is that we have a lot of pro-seq signal along each of these copies here and in some of these here. And it turns out it's all the chromosome 19 and chromosome 4 copies. What's even more interesting is that the signal that we see is not really indicative of a promoter, but is in fact indicative of an enhancer. So we think this is a very strong enhancer. It's very highly expressed and highly methylated. And so now we're hoping that scientists can use this as a way to understand what's actually happening in some of these cancer states when it may be destabilizing. But I want you to put a pin in this particular repeat because it's going to actually come back and be important in a minute. The other thing that we were able to do with the data that we had, because we had methylation data, we had sequence data for every single copy genome-wide, and pro-seq data for every single copy of every repeat genome-wide, is we were able to come up with maps that defined overall patterns of repeats. So for example, this is every ALU-Y sequence in the genome. I'm showing you the top 10% here. I'm showing you pro-seq signal. I'm showing you methylation signal. So we can look at exactly where CpG methylation happens in an ALU element and how that correlates with pro-seq signal. And what we identified for ALUs is we have a really nice promoter here. This is a known promoter in the first monomer, and then a really strong promoter, unexpectedly so, in the right monomer. This is a 7SL derived sequence, so it could be a signal of that. But what we know is that full-length copies are highly methylated and highly expressed, and as they become truncated, they increase in their divergence rates and decrease in their methylation, as you would expect. Deamination occurs at those CpG dinucleotides. The line signals are much clearer. If we look at L1HS elements, these are still hot in the genome. We have a beautiful promoter signal here, right at the 5' region. We have an antisense blip here, still don't know what that means. And beautiful CpG methylation. So very hot elements tend to have high methylation associated with their promoters. And that probably leads to this truncation that leads to the inactivation of these elements. And when you compare that to older elements, you can really see a difference in signal between hot line 1 elements and older inactive line 1 elements. They just look different. They're transcriptionally silent, they're truncated at the 5' end, they mostly lost their promoters. And so to sum up just a whole bunch of data, basically we were able to do high repeat diversity within human genomes, right? We were able to catalog that, identify new satellite arrays and composite elements, and we were able to look at transcriptional profiles of all of those TEs and correlate that with divergence and epigenetic features. And this serves as a reference for the community. We had some interesting findings with TEs and macrosatellites that act as promoters and enhancers, but more importantly, we're just providing methods and resources for the community. So, where does that leave us with the human reference? Remember I said it was an androgenetic line, but it actually had 2 X chromosomes. So that left the Y untouched. And the Y was a chromosome that was missing more than 60% of its sequence. And it's kind of an important chromosome, like 50% of the population has. So this just happened this year. Orang Marie from Adam Philippi's lab led this effort to sequence and assemble the full T to T Y chromosome. So we had to use a different cell line. We used HG002. It's a rare lineage found in Ashkenazi Jews and Europeans. And what the team did, Adam's team did, is supply 60x PacBio hi-fi reads, 30x for the Y only, 100 KB 90x coverage Oxford Nanopore long reads. So 90x coverage of everything 100 KB and over. So we were looking for really ultra long sequences. And Adam's team had developed a new tool for assembly called Virco that allowed for phasing, and we're using it for other genome assemblies now, but was really good at incorporating those Oxford Nanopore reads. And then polished it with, and there's no other word for it, just a shit ton of data, right? Like data you're not going to throw anything else, right? 250x Oxford Nanopore, 66x Illumina, 84x Hi-Fi, just multiple platform, lots and lots of data. And to just sort of sum up the entire project, here's the Y chromosome. And so if we look at HG38, this is what the Y looked like. There was this huge gap. And this huge gap existed because it was this really complex repeat structure, right? And what you can see here is we have these sign-derived HSATs that are arrayed next to another type of H satellite array, and they're just arrayed all over the place. And the way these graphs work, we love looking at these. These are called stained glass graphs. They were developed by Mitch Boulder from Evan Eichler's lab. The hotter the color, so when you have red, you actually trace back, that means this sequence here and this sequence here are virtually identical in sequence. All right, so it's a sequence identity plot. When you look at the centromere, that sequence identity plot looks a lot denser. That's pretty typical. You have these higher order arrays. We also have what are called centromere dip regions. I'll touch on those in a minute. But beautiful resolution. We've also been able to quantify all of the gene repeats. We've added 66% new protein coding sequence to the Y annotations, as well as more than 60% of the sequence. And this is really important because many of these genes are actually involved in human phenotypes that are of clinical relevance. For example, TSPY copy number is actually related to azoospermia. So, I just wanted to throw that out there. That paper just came out a couple months ago, and we're super excited about it. But really, I started this work wanting to look at transcription at centromeres, right? That's what I did not think I was going to be becoming a genome annotator. This is really what I wanted to do. But now we have this genome, we can actually do it. And so that was to ask the question, where are transposable elements in relation to these higher order arrays that exist at human centromeres, and do they have anything to do with centromere function? And so this is actually a figure from Nick Altimos using data from Gabby Hartley in our lab, where they had classified all of the satellites into the youngest satellite arrays, and this is that HOR structure, into the older satellites that have been sitting around for a long time. And right here is where the core of the centromere exists. So you can imagine this exists in a mirror form, right? And then into that, how often do we see hot elements? In this particular case, we're looking at lines. So line 1 HSs, we do see insert at a low frequency in the HORs. We never see old elements in those HORs. So these are always hot, recent insertions. The further away from that core that we get, the functional centromere, the older those TE insertions are. So then we wanted to combine that data with our transcriptional data, so our ProSeq precision run-on sequencing data, as well as RNA-seq data that have been developed by Megan Dennis and the team, and the epigenetic data that I was talking about from Ariel Gershman, taking those Oxford Nanopore calls and pulling out methylation calls, as well as looking at SEMPA distribution. So exactly where that centromere-specific histone actually lies in those repeat arrays, right? And we used cut-and-run data to do that. So I love this image. This is one of my favorite chromosomes. This is chromosome 19. And this is taking sort of a lot of this data. So this is the entire chromosome. If you look here, this is the methylation frequency along the entire chromosome, and looking at the CG frequency along the entire chromosome. This is looking at ProSeq positive and negative strand information. This is actually the centromere satellite annotation. So you can see this is the higher order array. That red is always a higher order array. And what you can see is the arms have lots of transcriptional signal, and most of that sits, on genes, as you would expect. We also have in the middle of that centromere, we can define exactly where SIMP-A nucleosomes incorporate. So even though we have a higher order repeat structure, SIMP-A nucleosomes only form at a subset of them, even though they're highly identical. So we can do that with cut and run data. We then correlate that with epigenetic data, and what we find is something that Ariel Gershman had identified first, is that SIMP-A domain correlates with a drop in methylation. Again, remember, all of these sequences are virtually identical. So this is not a sequence-defined feature, it's epigenetic. And she's called this the CDR, or the centromere dip region. Right, so it's a dip in CpG dinucleotide methylation that overlaps with SIMP-A occupancy. And if we look at the centromere as a whole, I don't see a lot of transcription there, right? Little blips here and there, but it certainly does not look like these arms. And so let's take a closer look at just this region. And so this is a zoom of just that. And you can see this is one of those stained glass structures that I was talking about. You can see really high sequence identity across all of these sequences. They're virtually 100% identical in sequence. And here, right here, is where we have the centromere dip region. So you can see this nice little dip, and then the rest of this, all this higher order array is highly methylated. And this is where SIMP-A nucleosomes actually are defined. And interestingly, you can actually see a little bit of a shift in sequence identity, right? So these are much more homogeneous than these are. However, these are the same as these over here, which do not incorporate SIMP-A. This is our repeat annotation, so you can see the satellites here. In green are TEs, and we can see beautiful pro-seq signal, including a satellite array of alpha satellite that sits here outside of the functional core, does not incorporate SIMP-A, does not have this weird methylation signal, and so we do see some transcription. Right, so it's not that satellites are incapable of being transcribed. So that's the first point. When we look under the CDR, however, we're getting very little signal, right? Very, very little signal. And so we really need to figure out, is that because of the computational restrictions that we have, or is that bona fide lack of signal at these particular sequences? One thing I do want to point out is one thing that we notice at this centromere and pretty much every single other centromere is what we call a TE boundary. And that is that surrounding the active HOR is a cluster of very recent TE insertions that are highly transcriptionally active. We see a lot of transcriptional signal that just surround this epigenetic feature that we have here. So we actually think TEs may be doing something important at defining the region, but perhaps not the SIMP-A location. So this is another centromere, just showing you sort of another track. We've got the centromere satellite repeats. This is a huge satellite array of HSATs. This is where the CDR is sitting. We got our drop in methylation, you can see very clearly there. If we look over here at that boundary, you can see some transcription of that HOR containing satellite. We have beautiful transcription at TE insertions. This is a hot line. This is a whole bunch of lines. This is actually what we call a TE island, which is reminiscent of the TE islands that define Drosophila centromeres. And we can track methylation signal all throughout. But when we look at the CDR, we get a very different picture, right? So here's our centromere dip region, beautiful methylation signal. And right here we have an embed. This is what we call a line 1 insertion. We think this is actually the boundary. We see transcription on the other side of it, but virtually none as we move through the centromere. There is a little. Like I want to be clear, there is transcription, just not a lot. So how can we actually tackle the problem that every reviewer is going to have, which is that pro-seq data is short read sequencing data, and we're mapping it to a genome that has many, many copies of the same short sequence. Because remember I said these were highly identical. So could it be that in fact, this is not a whole bunch of signal across here, but in fact is clustered in one section, right? But we've spanned it out because they're all the same. So it's just random mapping locations. So we decided to use the genome to not use the genome. And this is an approach that was developed in collaboration with Aaron Straight's lab, a postdoc Charles Lamousse, who decided, well, we have this beautiful annotation. Can we actually derive from that a database that gives us repeat-specific k-mers and centromer-specific k-mers? So we can actually look in raw read data and not have to map it at all. Right? So then we're no longer relying on bow tie, multi-mappers, or any of that. And that's exactly what he did. He developed this beautiful database. So if a k-mer was found in a particular read, he could say it's a line 1, or it's an allo-Y, or if it was an ambivalent k-mer, so maybe you find that k-mer in a couple of different sequences, it would classify it as such. So it would still narrow down what type of repeat it was. Right? So this was a beautiful database that he built. And we looked at both ProSeq and RNA-seq data. And coincidentally also did another method where we took all the raw reads and just repeat master annotated them, right? And got the same results. I think this process is a lot more quantitative. And so what did we find? Well, what we find is that if we look at CHM13 ProSeq data and CHM13 RNA-seq data, we can track all the different, quantities of repeats and their transcription levels. So this is reads, right? So this is how actively transcribed they are. And then we decided, well, we don't want to make any general statements about transcription at centromeres without looking at some other data types. Because this is a molar pregnancy, we should be looking at some maybe embryonic stem cells, ileum, and maybe a terminally differentiated cell line. So we did that. We took all these different cell lines and did the same thing. And you can see the frequencies are fairly stable. What you can see very clearly is that RNA-seq does a really poor job of picking up active transcription of repeats. ProSeq is much more accurate. But what you'll see is way down here at the end, and we have to zoom into that, are very few satellites. Right? And that's what we're looking at here. CHM13 is here in red, so we have 0.8,000, 800 satellites that are actually giving us any signal of ProSeq data, right? So we know there is some RNA polymerase occupancy that's just very limited. So then we thought, well, most of the cells that we're looking at are in interphase. Maybe this is a cell cycle dependent thing. So SEMPE is actually incorporated at one point in the cell cycle, and that's late telophase, early G1. It's a very dynamic process. It has to happen every cell cycle. So we took cells and in collaboration with Leighton Core's lab, which was just down the hall, synchronized them and released them. And so we have all cells going through all stages of mitosis and then right after. And did the same thing. Did ProSeq and annotated every single repeat in k-mer we could find in that short read data. So this is all actually using HeLa cells. And what you can see here is we have lots of signs that are expressed in asynchronous cells. So these are sitting in interphase. It's pretty exciting. But when we look at mitosis, there is still transcription at mitosis. It's just very limited. If we look at satellites, which is this gold thing here, virtually all of it is off at mitosis. There's a little bit, but most of it actually happens after release from mitosis. The things that are very highly transcribed, we actually see some LTRs that are very highly transcribed, and some tRNAs that just go right up at mitosis, which is really interesting. But the end result is that centromer transcription happens, it's just very limited. All right, so for human centromeres, we know we have emerging TE clusters and embeds within those higher order repeat arrays, but not in the SEMPE domain. Not a single TE was found in any CDR at all. Right? And so we can also date each of those TE insertions because now we have a complete compendium of all the human TEs. We know that satellite transcription is detectable but very low in that SEMPE domain, but we know that TE transcription does mark boundaries of where SEMPE will find an HOR. So we think they may be a more superorder structure rather than defining the actual loading process. And satellites can be transcribed. If they're outside of the centromeric chromatin, we do see transcription from them, which is still a mystery. So that's sort of a lot of human data, right? So now we know a lot about human repeats. We know quite a bit about transcription and where transcription happens in satellites. What does this have to do with centromere forms in other species, right? So we have our dominant satellite forms. We see those in human, we see them in mouse. We can get neocentromere forms even in human clinical cases, right? And so we've got some work that we're doing on that, but I'm not going to talk about it because I want to talk about some cool things in the last few minutes that I have. And that is this species group here, which are the gibbons. Some species, the Hulak gibbon in particular, has a complex centromere, whereas its sister taxa, the Damascus gibbon, actually has higher order repeat arrays that contain alpha satellites. Really great comparative model. So what does that look like? So this was actually the thesis work of Gabby Hartley, who just defended this summer, who was asking, does the activity of transposable elements actually impact chromosome evolution in any way? And why this particular group of primates is so exciting, they are a lesser ape. So they're our most distant related apes, right? So they are not a monkey. Do not ever call them a monkey. There are 16 different species. All 16 are critically endangered. So they are losing their habitat. They're actually a fascinating organism. If you ever want to learn more about them, they're the only thing outside of humans that can actually sing operatic tones. You can get some of these videos online. They're incredible. But why we love them is their chromosomes are all sorts of fucked up. And it's great. So in a very rapid and recent radiation, we see massive changes in karyotypes. So we have a diploid number of 38 up here, we have a diploid number of 50 here, a diploid number of 52 here, but not just the chromosome number has changed, but the chromosome forms. So each of these blocks are the human syntenic region, and what you can see, you don't need to be a cytogeneticist, you can be a kid playing with Lego blocks, you can see that each of these chromosomes is different. Right? Massive chromosome restructuring has happened in a very short period of time. And so we were really interested in this guy here, the Hulak, because we knew quite a bit about the centromeres thanks to our collaborator on this project, Lucia Carbone. And we knew that there were no alpha satellites in any of these centromeres. Remember, this sister taxa down here has alpha satellites at every centromere. So all of these centromeres are new. If they have no centromeres, what are they? Well, we knew 4 were what we'll call ENCs, evolutionary new centromeres, and no other species had them at this location. So it's kind of like a neocentromere. And we also knew from work that she had done that a new composite retrovirus had evolved to take over the centromeres of many of the chromosomes. And that's what I'm showing you here with these bars. So some of these ENCs, 2 of them, had this lava sequence at them. But some didn't. And in fact, we found through genome sequencing that 2 of the centromeres that hadn't been annotated had this guy here, SST1. So remember that? That's from an earlier part of the talk. That's this guy here, where we have copies all over the human genome, very active copies on chromosome 19 and chromosome 4. And it turns out that chromosome 19, that is the location of this centromere in this particular species. So that's pretty exciting. But what exists at 2 of the other centromeres that we have no idea what they are? This one here on chromosome 11, and this one here on 17. So she started her PhD, like all good students, I'm going to do a whole genome, and it's going to be great. And the technology continued to change. And so she did this genome probably 100 times. But we have a genome assembly now that is in single chromosome contigs. And its gene completeness is very high, and it's what we call a sub-T to T genome. We've actually captured about 11 of the centromeres of this genome. So it's almost complete. We did capture the centromere of one of these ENCs, chromosome 17, and by using functional annotations, using cut and run and ChIP-seq data for SNP-A, B, and C, so these centromere proteins, we're able to actually really refine the functional regions of these centromeres. And in doing so, we looked at our repeat massacre track. So this is all different kinds of repeats. This is SNP-A, B, and C. Really nice. Centromere looks great. But this looks kind of, wow. I see a repetitive structure here. You guys see that? right? Looks repetitive, right? This little block here looks like this, looks like this, looks like this. This is what we call a composite sequence, right? We fished it to make sure we were looking at the right chromosome. We are. And we took a deeper dive and looked at each of those composites. So it's a multi-composite array. We now have cut and run data to fully refine what happens on each of the repeats within this array. So each block consists of many different repeats. Most are old, right? We've got an ALU-Y here that's fairly recent, but ALU-SX, very old, L1ME, old, right? So these are old repeats. This is not a new active repetitive insertion. It's some block of sequence that has now been repeated. And it's been repeated a bunch of times just in this genome. When we look at Damascus, which has alpha satellites in the centromere, they have this on a euchromatic arm in one copy. And in fact, they just have alpha satellites. This thing is not repetitive at all. So if we just looked at the Damascus genome, this would never have been classified as a repetitive sequence, right? When we look across the phylogeny, we look at some of our other genomes that we have coming off the block for some gibbons, like the Hylobates. We just have one copy. And in fact, if we look at all the primates, including human, there's just one copy. Right? So even in human, this is not annotated as a particular repeat. It is a repeat in gibbons, and it forms the active centromere. So this raises the question that we have in the annotation field: how do we annotate this? This block is conserved as a block across many millions of years of evolution, and then it's tandemly arrayed in one species. So how do we score that in the human genome so that if somebody sees that's arrayed in a clinical case, there may be some information there that's relevant? So we're pretty excited about that. So another group in the last 2 minutes that I have are obviously the wallabies. We always have to look at the wallabies. Not just because they get to take us to Australia, but they also have fascinating chromosomes. They've undergone rapid chromosome radiation in some species, the kangaroos and wallabies, for example, whereas other marsupial species like the dasyurids have completely stable karyotypes. And we don't know why that dichotomy exists. And that's one of the things that we're looking at. So the Tamra wallaby, which is one of our reference genomes, has a sister taxa, the redneck wallaby. You can see this nice little red neck that actually has giant HOR containing chromosomes for centromeres, right? So we see this same juxtaposition that we see in other species. So Patrick Grady actually started this project at the start of the pandemic, and it turned out to be a great thing to do, right? What are you going to do in a pandemic? You're going to build genomes because you can't go in the lab. Why study the Tamra wallaby? So I want to give you guys a piece of biology because I think that's important. The Tamra wallaby and all marsupials are very interesting to study early development. They've been an early developmental model system for decades, over 30 or 40 years now. And that's because they give birth to what are called altricial young. So this is actually a newborn kangaroo. And what you'll see here is he's got very tiny little back feet, shocking considering how big those feet become. Very well structured front claws. He's got a complete mouth, olfactory system. Eyes don't work, but the nose does. And so this little guy, post-birth, will actually climb up into the pouch, attach to a teat, and progress through development. And so here you can see this is him here, his little tail, his little feet, his front claws, right? And he's going up and he's attaching to the teat here. Marsupials also do this really fascinating thing where they'll have multiple teats in a pouch producing multiple kinds of milk. So they'll have this little pouch young newborn getting a protein-rich milk, and then an older kangaroo that'll be jumping in and out of the pouch getting a very sugary rich milk. Right? So it's a really interesting study model system for lactation. Because of this early developmental birth, this altricial birth, sex differentiation happens after birth. So this is a process that in mouse and humans happens in utero. So it's a really great model system to study sex differentiation and defects therein. And that's what it's been used for. It's the reason we discovered that SRY is actually the sex determining gene in mammals, is that it's conserved on the tiny little Y chromosome of the species. So we, being genomicists, wanted to make a genome assembly. And so we did. And we used mostly ultra-long sequencing data to do this. So this is an ONT only assembly, and we're able to find every single chromosome. We can define all the things that were missing in an earlier version of this genome assembly that came out in 2011. Most of the genome was missing, as you can see. We can also define new repeat annotations. We've annotated 26,000 new genes and define repeat diversity. And so we're pretty excited about this. What we've also been able to define are every single centromere. So we've sequenced every single one to complete coverage. And what you can see here is something very different than what you see in human. We don't see these giant red blocks. We see a few that have them here and here, but then we see some that are not really highly repetitive at all. Right? So really complex centromeres, and we're diving into that now, and it's very different than a human centromere that has an HOR. We've expanded this study to a variety of different marsupial species. We now have 2 tamer genome assemblies, one that's a male. We have a swamp wallaby assembly. We now have a redneck wallaby assembly and 4 other wallabies. And we are using that to compare to the dasyurids, which have very slow chromosome change. Right? Remember that comparative model that I talked about, including the Tasmanian devil, the spotted pole, and the fat-tailed dunner. And this is the difference, right? So you can see this very clearly when you just do a synteny map. These are 2 kangaroo species. They diverged about 2 million years ago. You can see massive chromosome rearrangements. When you compare them to 3 different dasyurids that differentiated or diverged 20 million years ago, they're all pretty much the same, right? So very different timelines of chromosome change. And when we compare each of those genomes, we find really cool breaks of synteny. So this one's very obvious here. You can see this break here between the fat-tailed dunner and the tamer. All of these tend to be centromeres. Right? So centromeres are hotspots of chromosome evolution in this particular species. And I'm going to end this talk with one little tidbit of what we have discovered that's quite fascinating, is that each of those centromeres contains not a satellite sequence, but another repetitive sequence. Do you guys recognize what that is? Anybody?
T7 promoter.
Telomeres. T7 promoter. That's me. So this is a telomere array in the middle of the chromosome, and it's actually acting as the centromere. We validated that with ChIP-seq and now cut and run data. We still have low methylation across this region. And it's interesting because we can trace this back to what is actually the telomere in the dasyurid. Right? So somehow the lability of these chromosomes, they're able to use telomeric sequences as centromeres and basically flip-flop chromosome arms around. Right? So it's a pretty exciting way of actually repurposing a repeat in your genome. So hopefully I've impressed upon you that T to T scale genomes are important. You can identify functional features of genes, regulatory regions, and particularly repeats. And more importantly, we can start looking at evolutionary breakpoints, centromeres, and regions of instability. We can even look at how new species may form. And this is just a snapshot of some of the species. We've just started working on the T to T ruminants, where we're doing 69 different ruminant species with Tim Smith's group and many other species. And so I believe it was sort of Adam Phillippe's tweet, we just need to sequence all the things and we'll be fine. And just to close out, we're actually working now on doing some extinct genomes, and we just had a really exciting result literally yesterday where we had a 10 kb read from an extinct genome sample that was taken from a museum, so we're really excited about that. But really, thanks to people in the lab, Jessica Storer, Savannah, Gabby, and Patrick, who were really leading that T to T effort in the lab. Adam and Karen, who created an open and collaborative environment and supported trainees to have really cool ideas and to follow them out. It was an extraordinary experience, and I hope more science is done that way. And then collaborators on many of our other projects, and I'll leave it there.
Questions? Nelson?
So that was fantastic. When you talk about measuring transposon transcription, you have this novel Kamer method that doesn't even use the new T to T genome assembly. So what is the recommended way then to measure transcription, especially with comparing control to a disease sample?
Well, it depends on what you're looking for, right? So it did use the T2T assembly in that we were only able to make that K-mer database because we had the T2T assembly. Without that, it would have been an incomplete database completely. We would have been looking at about 80% of the repeats. And so now we can look at every SNP, every variant of every copy of every repeat in that one genome, right? So that is just CHM13. What we're probably going to find is that each human genome is kind of a little different, right? And so when the Human Pan Reference Consortium comes out, it's going to be a challenge to track those repeats. But I think that's what we're excited about tackling in the future. If you're looking at things like a knockdown, gene on, gene off, you know, if you know what you're looking for, we actually found that the mapping data pretty much mapped up with the K-mer data. But we had to map a variety of ways, right? So we had to map and do what's called K-mer filtering. So one of the beauties of the TDT genome is that there is a K-mer file of every unique K-mer in the human genome. And so you can map and then filter, only show reads that have a unique K-mer. So you know that read came from just that one spot in the genome and nowhere else.
And so this would take care of transposons in those arrays that a straight up location specific mapping approach might miss?
Correct.
Okay.
Correct. And that's why those orthogonal approaches were developed as part of this work. Right. So there are multiple, right. So there's not one way to do it, unfortunately, but there are a couple of really great creative ways that use the T2T reference, the K-mer database that is unique K-mers and the K-mer database that is every repeat. Right. So you've got two different K-mer databases there.
Sean?
Yeah, you had mentioned these acro repeats of it that, correct me if I'm wrong, they contain protein coding?
The acro doesn't, but one of the other composites does. Okay. Yes, there are actually several that do.
Are those interspersed with the rDNA repeats?
No.
Or their boundaries, they're at the same genomic locus, just...
So the acros, there are a few that exist on the rDNA containing chromosomes, the acrocentric chromosomes in human. They're not actually right next to the rDNA arrays. Some of the SST1 arrays are, interestingly enough. And I think a paper just came out in bioRxiv, it may have actually been published now, from Eric Garrison's lab, that showed SST1 is actually the fusion site in Robertsonian fusions of those acrocentrics, which is kind of like, wow, okay. We saw that in evolution and now we're seeing it in human clinical samples. It's pretty exciting. Where we see the ones that contain genes, the vast majority of them are actually on the X chromosome. Not all of them, but many of them are on the X. So the X is sort of a harbor for multi-gene families for some reason.
Thank you very much for an interesting talk. So I have a question regarding the annotation of the regions which have reached the repeat. So when I play with the programs like the Repet, I think it was this program that allows the annotation of this region. What we get is a lot of redundancy and intersection between the annotation of a particular region. So basically, there are big elements annotated, small elements inside of them. There's intersection, so it looks like a chaotic thing. So how do you resolve this problem that one entity is one entity, another entity is separated?
Is that, are you using Repeat Masker to do that?
I think it was a Repet program.
Okay, so that's a different program. So Repeat Masker actually uses consensus and Smith-Waterman scores. It's a little bit more accurate. But what it comes down to is what database you're using. So for Repet, you still want to use the DFAM database. So one of the problems in the repeat field, and there's a consortium of people that are actually getting together to try to solve this problem, is there are many different ways to find repeats, and there are many different ways to classify them, and then there are many different databases. Right, so a lot of data went into RepBase. Everybody heard of RepBase? RepBase is now behind a paywall, and it is not updated. So none of the CHM13 data is in there, and it has not been updated for years. And so that's a problem, right? So DFAM has been updated for human, but not for anything else, really. There are a bunch of data in there, and we're trying to, everybody's trying to clarify that. Another issue is how Repeat Masker works. There are so many different parameters that will impact how your Repeat Masker calls are done. And Aaron Smith, who developed the program, has yet to publish that compendium. I'm lucky enough that Jessica works in the lab and knows all of that, and we're happy to share it with anybody. She will tell anybody, anywhere, anytime how to do repeat annotations, and the things you have to watch out for. But it is kind of a problem we're hoping to bring the community together for just that reason.
In the back.
So this kind of this chicken and egg question, right? How, what's the relationship between centromere diversification and evolution and speciation? Which comes first? And what's that telling us about the underlying mechanisms involved in mitosis that kind of putting the pressure on the systems of selection and re-evolution?
The answer to that is yes. So it's a complicated question because in some species, centromeres have nothing to do with speciation, right? Dasyurids, for example, that's probably ecological adaptation and environment. For the wallabies, centromeres have because of this telomere repurposing, and that somehow that allows them. There's actually more to the story. There's the turf pathway is slightly modified in that particular group of organisms, but for whatever reason, when we get hybrids between many of these species, many of them are viable and they have new chromosome rearrangements. And so that is combined also with an ecology framework. So for example, rock wallabies, there are 21 species, 20 different karyotypes. And that's probably because you get a hybrid, it goes hops on a new rock, makes a new breeding colony, and it's pretty isolated. Right, so you can start to see things that in a big population may be heterotypically negatively selected against become neutral or even positive. So that's why I say it's kind of yes to all of the answers, right? Because we don't know. So, is a centromere shift to a new location enough to make a new species? Well, in humans, when we see a centromere shift to a new location, that's usually pretty bad. We see that in clinical cases. And it's associated with other chromosome aberrations. In many cases, we see marker chromosomes in human. We only see them in 20 or 30% of the cells, developmental delay, really devastating consequences mitotically. And many of those individuals don't actually live to puberty, so we don't really know how far meiosis goes. So it is a fascinating question. And, we have a chromosome difference between us and orangutan. So clearly that got fixed at some point, and speciation happened. Was it the rearrangement that drove it, or was it the environment and the rearrangement happened independently? I think we're seeing both. So I think some groups like gibbons have a more unstable genome that promotes chromosome rearrangements in an ecological adaptation scenario. I think wallabies are the same. Horses are the same. So for those of you who don't know, there are many species of horses, and they all have different karyotypes, very rapidly evolving chromosomes. So again, totally different than cats. Many species of cats, they're all pretty stable in terms of big chromosome blocks. Where T2T data is going to come in useful is now we can start looking very refined, right? We were looking at chromosome segments that were thousands of megabases, right? And so, or hundreds of megabases. Now we can actually go to each nucleotide and say, oh, actually there is a small difference between the lion and the tiger. There's a chromosome inversion that has TEs around it or whatever. And so I think we can start to really address those questions now for the first time. So it's exciting.
Also, a question about transposon expression. Do you think now with the new T2T assemblies, will males have higher expression of transposon RNAs compared to females because of the load of repeats on the Y?
Well, it's interesting that you say that. So one of the things that we've discovered in the Tamara wallaby reference is the Y is actually a harbor for full-length TEs. There is a higher density of full-length active TEs that sit on the Y chromosome more than any other chromosome. And the reason this is remarkable is unlike human, which is a highly repetitive chromosome, the Tamara wallaby Y chromosome is only 3 megabases. It's really tiny. So if we normalize for length, this is, it really fits into that, toxic Y hypothesis. And so we do think that could be the case for Y-born genes or Y-born repeats. How sex differences impact transcription of TEs across the rest of the karyotype, I don't think anybody's looked at yet. At least not in mammals, in a way that it's based on new T to T assemblies. I mean, we have that early mouse data, but that data is 15 years old and was mapped to a genome that was maybe 40% complete. So I think there's a lot of data that's sitting out there that just needs to be remapped to new genome assemblies, and I think we may discover some really interesting stuff. A lot of that data. So if there's another lockdown, you get bored, remap.
Don't say that.
Okay, if there are no other questions, let's thank Rachel.
I'll be
Oh,
hanging.
one.
Thanks.